The overall goal of this study is to introduce two flow cytometry-based immunological assays for measuring the cell-mediated immune response to West Nile virus infection. In the in vitro T-cell priming assay, dendritic cells, or DCs, purified from West Nile virus-infected mice are co-cultured with naive CFSE-labeled T-cells purified from OT2 transgenic mice in the presence of ovapeptide. The proliferation response of the T-cells is then analyzed by flow cytometry in a biosafety level 2 laboratory. In the modified intracellular cytokine staining assay, splenocytes from West Nile virus-infected mice are stimulated ex vivo with West Nile-specific peptides in a microcentrifuge tube. And then, the cells are stained for intracellular cytokine production and analyzed in the tube by flow cytometry in the biosafety level 2 laboratory. Ultimately, these methods can be used to examine the role of specific signaling pathways in the activation of T-cells in West Nile virus-infected animals, and they significantly reduce the performance time inside biosafety level 3 facilities.
Though this method can provide insight into West Nile virus cellular immunity, it can also be applied to other biosafety level 3 pathogens.
For the in vitro T-cell priming assay, begin by isolating splenic dendritic cells from wild-type C57BL6 and Myd88 knockout mice inoculated with West Nile virus, and control uninfected animals with anti-C.